at the next session of Natural Sciences. We have two speakers, and the first one is Igor Grochowina, and the topic is investigating the molecular mechanism of Hsp70 gene domain interaction.
Hello everyone. Could we put the lights off? Could I ask? Thanks. Thank you very much. Okay, so as you probably heard before, my name is Igor Grochowina and I'm from Laboratory of Evolutionary Biochemistry of Intercollegiate Faculty of Biotechnology of University of Gdańsk and Medical University of Gdańsk and blah blah blah. And in our lab, so it's evolutionary biochemistry, but we use evolution. Evolution is not the target, the main target of our research. It's rather a tool to investigate the basic mechanisms that rule the protein-protein interactions in the cell. So let's start with the sentence that most of you probably had more than once in your lifetime, probably in high school. Mitochondria are the powerhouse of the cell. So actually there's much more going on in mitochondria than that. For example, some biosynthesis of iron sulfur clusters that are ancient prosthetic group that are critical for the survival of the cell, and they perform many functions. So in mitochondria they are involved in Krebs cycle, oxidative phosphorylation, such as this aconitase with 4-iron-4s iron sulfur cluster. Bound by cysteines. In cytosol, it's the tRNA metabolism translation, nucleus DNA replication, and disorders in iron sulfur cluster biosynthesis are related to many diseases like Friedreich's ataxia, sideroblastic anemia, and so on and so on. So the iron sulfur clusters are built on the isoprotein, which is a molecular scaffold, and they are synthesized by the cysteine, the sulfurase, so the sulfur donor and the iron donor YFH1. And the next step is actually the Hsp70 chaperone-mediated transfer to the recipient protein. So, yeah, and about the Hsp70 chaperones. These are the conserved family of proteins that are present in practically every single taxa, and every single organism has numerous Hsp70 chaperone proteins that their main function is heat shock response, also protein folding, transport through membranes, collagen coat disassembly, and also iron sulfur cluster biosynthesis. And these all activities make them, for example, really useful in cancer cells. So cancer cells rely heavily on Hsp70 proteins to deal with unfolded proteins and such on. So it's really interesting that all these functions depend on a single mechanism. That is the J-protein mediated substrate binding and hydrolysis of ATP that refolds the substrate to some extent, and for example it can unfold the misfolded protein a bit, and it can perform its function again, or it can transfer the iron sulfur cluster to the target protein. So the J proteins are also a quite conserved family. They are characterized by the J domain, which are HPD motif. On this loop you can see here, they do two things. They deliver the substrate to Hsp70 with the C-terminal domain, and also the HPD motif stimulates the ATPase activity of Hsp70. So it's a regulator of its activity. It's critical for its substrate specificity and overall activity. So for example, in yeast mitochondria we have two Hsp70s and three J proteins. Each one of them has different function, import to mitochondria, refolding, iron sulfur biosynthesis, and some Hsp70 J protein pairs are specific, while some of them are not. So you can think about J protein as a guide dog for Hsp70, because Hsp70 doesn't really care what it binds. It requires a J protein as a targetase to to bind the protein. So, our main question in our research was, what's the mechanism of this interaction? Because that's kind of unclear at the moment. So this interaction over there, we wanted to investigate it. But how? How do we get to that? The first obvious response to that question is get a crystal structure. But there are several problems. There are some modifications required in the sequence of both proteins to stabilize the interaction. Also, in crystal, we observe no conformational dynamics. There's one stable structure. And if you want to investigate mechanisms, it's difficult to do that while you can't really get a crystal without any modifications that can disrupt the interaction you want to investigate. So we used molecular dynamics simulations. So the theoretical approach, but as I will show you in a while, it works well with biochemical results too. So we can really rely on them in our research. So the basic mechanism, so how do we perform molecular dynamics simulations is we know the initial positions of atoms, calculate the forces between them, and then simply solve the Newtonian equations of motion. There's lots of mathematics involved, but I've hidden them under the text and under pictures, so the mathematics is here only for my personal enjoyment. And there's a force field that simulates the physical system, so electrostatic and van der Waals interactions, all that kind of stuff. And as in the Gogol Bordello song, oi oi oi, accelerate the protons. So we took two approaches to build our structural model. First, we sampled 23 conformations of the complex, which summed up to 21 microseconds of the trajectory. So there are several trajectories. They just shook a bit, and they converge into a cluster of bound J protein to the Hsp70. And we are lucky enough that the half of the overall trajectory was our conformational ensemble, and that's the crystal, that's the central structure of this ensemble. The second approach was to spontaneously observe the binding of this protein, because we thought that if we let it just dock onto itself in molecular dynamics simulations, we can observe the real place of binding. So we've done a few simulations, and the binding time was really short, up to 30 nanoseconds. It's not really much. And the structures obtained were really similar. I'll show you how it looks like. So like this, we can see the J protein docking onto its binding space, binding region on the Hsp70 protein. So the J domain docks onto the nucleotide binding domain of Hsp70, this gray part, and that's how we obtain the model. It looks like this in kind of real time, but in terms of nanoseconds. So here's our structural model. That's the substrate binding domain, nucleotide binding domain, and the J protein bound here. The HPD loop packs tightly at the interface between these domains. Do I speak loudly enough? Yes. Okay, thanks. So we wanted to investigate what are the forces, what's the mechanism of interaction. So first, the recognition. We've calculated the electrostatic potential around the proteins, and we've seen that there's large positive charge over here and large negative charge over here. So the obvious thought that you might have is that they will bind. And that's exactly the case here. So we can see that helices 2 and 3 of J domain are involved in the recognition interface between these two proteins. So they pack tightly onto the nucleotide binding domain of the Hsp70. So we confirmed it with biochemical ATPase assays. So we measured the ability to hydrolyze ATP by these proteins. And we've seen that if we mutate these residues, we see almost no interaction. So we performed also some simulation. We also performed some simulations that showed that two substitutions, two alanines, are sufficient to completely negate the interaction. So no affinity here. So we can see that this recognition interface is important. Do I have much time left? Okay. So, but the real question is what happens next? we can see that the HPD loop of the J domain has conformational flexibility, but actually only the bound conformations, these two, are energetically favored, so they have low free energy, so they are more probable to occur. So we can see that the bound state is energetically favorable. So that's nice to know, but what happens next? The one important thing we have to know about the Hsp70 cycle is that there's an interdomain buckle between the linker NBD and the SBD, and these three residues over there. So this buckle can spontaneously break, but we see that in presence of J protein, we actually see more unbuckled state. So this interdomain communication can be easily broken, but there's also a question how it occurs. So the D50 of the HPD loop is a conserved motif, highly conserved. As we see, it steals the arginine 207 from its native partners, as depicted here. That's here you can see the free energy profile of on the distance difference between these four residues. So you can see the distance between native partners and arginine between D50 of J protein. And we can see here that the D50 bound state is energetically favorable, so we can see that D50 can steal the arginine 207. So the next question, it's kind of obvious, but it has to have some large-scale effect on proteins, or maybe it doesn't. We checked that, and we've seen that the domain undocking of Hsp70, which is a critical step in the ATPase cycle, so it's necessary for it to function, the undocking of the domains is easier when the J protein, so also called Hsp20, is present. So, and we also seen that the hydrogen bonds between the alpha helical lead of the Hsp70 and the rest of the protein, they break. They are less frequent, so it facilitates the lead dissociation, and lead can close on the substrate, stabilizing the binding. So it works a bit like the Venus flytrap. So it's like closes on the substrate protein and doesn't let go. Evolutionary biochemistry, right? We've done some evolution stuff on that, and we've seen that when comparing the sequences of J proteins from different species, we've seen that in bacteria this sequence is conserved. But we've seen it for two proteins, both of them, Hsp70 and the J protein. But in all the eukaryotes, there's no clear conservation, so this mechanism we can see is not conserved. So then we can actually think about it as a separate mechanism. So, in conclusion, we can differentiate between two parts of the interaction. So first, the variable interface between the J domain and Hsp70 protein that changes in evolution and determines the specificity of binding. So the multifunctional Hsp70s can have less defined recognition motif, but the mechanism of stimulation of ATPase activity, so the trigger, is evolutionarily conserved, and it acts through interaction of J protein HPD motif with arginine 207. So now we know how this interaction works, so we might design some inhibitors that can be applied in medicine, such as cancer research and such, also Friedreich's ataxia. So it's not so useless as it looks like. So I'd like to thank my collaborators from University of Gdańsk. You can see these merry faces over there. Also from Technical University of Gdańsk, these guys do the mathematics, I do it with them. And also from the Department of Biochemistry from Wisconsin-Madison. So, thank you very much.
time. Is there any questions? Okay.
Could you go back a few slides, maybe?
Sure.
To this conservation, evolutionary conservation, we can clearly see that between E. coli and Saccharomyces, there is clear conservation. Do you have any knowledge how it's so? It's you know,
Convergent evolution. Convergent evolution is actually interesting because DnaJ is there are two J proteins in mitochondria of yeast, and also there's one protein in E. coli that is responsible for iron sulfur cluster biosynthesis. That's DnaJ, and by duplication it occurred. There was another HSCA, and the DnaJ is actually similar to the multifunctional J protein, like the J protein from multifunctional Hsp70, but it shares sequence similarity with the yeast system, which we think is convergent evolution. So the recognition is conserved, but the affinity is lower because of the loop in bacteria is a bit longer, so the interaction is between arginine and the aspartic acid is not so easy.
More questions? Someone?
You have shown nice interactions between those proteins, and do you have any confirmation of the functionality of those interactions, for example, by using specific mutants which disrupts the interactions? Or?
Yeah, here the biochemical, here the in vitro experiment, so it's in vitro ATPase assay, when we have purified proteins, and we see that the mutations of these residues are critical for interaction. So we can see the ATP hydrolysis rate is significantly lower in this case. We also done some in vivo tests, but they are fresh, so they require additional.
This was my second question, but you about the in vivo.
Yeah, so we done
So
in vivo.
they're ongoing.
We see pretty much the same result, but we are waiting for for repetitions to ensure the statistical significance.
Okay, thank you.
I have one more question. Thank you. Somehow this effect is more pronounced. It looks like it's more pronounced in Hsp70 mutants. Do you have any comment on that? Why is that mutations in J protein?
I would say it's comparable. That we can. I mean, it depends on the residue you mutate, because this interface is kind of mobile, so it all goes to the electrostatic potential around these regions. So I don't see the reason why could they be lower. They are a bit lower, but not as much that we could be somehow concerned about it. So we can see that it makes the interaction less probable. So that's it. And it's residue specific. We can see that, for example, the K70 residue, which is on helix 3, which is a bit more distant than the helix 2, has less input to the binding than the rest of the residues on the J protein. But why there's a slight difference, I can't say for sure. I simply don't know that.
Okay, so thank you very much.
We would like to invite next speaker. I would like to introduce our next speaker, Mr. Rafał Ostrowski, with a presentation entitled Quo Vadis Antibiotics, Can Murine Hydrolases Replace You?
Thank you for presenting. I only add that I'm PhD student on Department of Applied Microbiology, University of Warsaw. And why I ask this question in my title of my presentation, because like maybe you know that first antibiotic was discovered by Alexander Fleming in 1928, and first resistant bacteria was discovered was identified 4 years after the penicillin was when mass production of penicillin was started. And now we have a very serious problem with antibiotic resistance, and more details about this topic you will listen in the next speaker, or my colleague from university. And my second question is, are we living in the post-antibiotic era? This is very serious problem. We can just look on the popular website of and headlines of popular website. For example, the Supreme Audit Office alarm 400,000 infected people in hospital. Superbugs discovered on Pomerania. Superbugs in hospital. This is from May this year, January this year, and May 2 years ago. So this problem is very serious. Third question. Extra information that Jim O'Neill, the British economist, to order to British government calculated in 2050 years on the problem with infection caused by the antimicrobial resistance bacteria will be die 10 million people per year, more than cancer. So, my third question is, do we have any alternatives, any another solution? Yes, of course we have. One of them is the new classes of antibiotics. We can use bacteriophages to treat the infection, antimicrobial peptides, or murine hydrolases. And the last example is my favorite, because during my PhD study I'm investigating some role of in physiology in bacterial some of the murine hydrolases. And today I would like to show you some example, some idea, some concept how we can use murine hydrolases to fight with bacteria. And I would like to add that every topic is only concept. Any clinical trial were done, maybe it, but especially on the last example. And for some definition for people who don't know what is it. Murine hydrolases is the class of enzyme which cut bonds in murine, and they are involved in several crucial processes in physiology of bacterial cell, but they are also appeared in animals, for example in human. Lysosomes are part of innate immune system, and it's we can find it in saliva and tears. And murine hydrolases also appear in bacteriophages, these viruses which infect bacteria, and bacteriophages use them to break the host cell in the last stage out of infection. And here we have a comparison of two types of bacteria. Gram-positive bacteria has on the yellow color plasma membrane and thin layer, multi-layer murine cell wall. Gram-negative bacteria has also the outer membrane, and this is problem for using the murine hydrolases, because the outer membrane, the enzyme cannot access to their substrates to the murrain. So the murrain hydrolysis is less effective against gram-negative bacteria. And we can define murrain hydrolysis by origin, lysosome and eukaryotic bacteria, and virus-related murrain hydrolysis. And the first example of using the maybe some ideas is this one from eukaryotic origin. We can use lysozyme like a food biopreservant in case of wine, beer, smoked fish, or cheese, and another. And another more complicated example is that we can make transgenic animals, like, for example, DnaJ is the little laboratory fish, the model organism. And this transgenic DnaJ with the gene and condylysosome increase its resistance to fish pathogen like Flavobacterium columnare or Edwardiella tarda. Another example of transgenic animals is the transgenic goat, which produce human lysosome into their milk, and this milk inhibits growth of Escherichia coli, Staphylococcus aureus, or Pseudomonas fragi. And also this milk can reduce diarrhea in young pig infected with E.coli DnaJ, and these pigs dehydrated more slowly. Symptoms of intestinal inflammation were weaker. The pigs returned to normal leukocyte proportion in blood faster, and also they showed less damage in the intestinal villi. Second class, like I said, is the hydrolytic bacteria, and the most interesting is lysostaphin. Lysostaphin is the enzyme produced by Streptococcus simulans, and it can be used like antimicrobial agents against Staphylococcus aureus. Next example of transgenic animals, transgenic cow, which produce lysostaphin into milk, and this kind of cow is more resistant to the mastitis. Mastitis is the serious cow disease. For example, in United States, all costs connected with the mastitis is on the level $2.5 billion USD. And two additional facts: this lysostaphin also stay in the cheese during the cheese production, and here you have the level of the percent to the previous amount of lysostaphin in this particular stages. It's thanks to this lysostaphin, this cheese, it can be the microorganism, for example, this Staphylococcus aureus won't be grow in this kind of cheese. And also lysostaphin is produced by lactic bacteria. Scientists made this kind of GMM microorganism, and supernatant of lactic bacteria culture reduced the number of methicillin-resistant Staphylococcus aureus 8-fold in comparison to the control. And the last classes of the murrain hydrolysis is the virus-related murrain hydrolysis, and some example, for example, and just in the 1993, very early, scientists made the transgenic potato with the murrain hydrolysis from phage T4, and this potato was stayed more resistant to the plant pathogen Erwinia carotovora. The PLYC from phage C1 is over 100 times stronger than other known murrain hydrolysis, and especially is very effective against horse pathogen Streptococcus equi, and it's proposed to use this enzyme as a disinfectant aerosol to sterilize the box for horses. And some other fact that PLC is 1,000 times stronger than the current using agent Virkun S. And another experiment when PLYC was administered orally to mice before infection with Streptococcus, only 500 units of enzyme. In this amount of enzyme, no bacteria was detected after this mice section. And very, very interesting example is the artilysins. It means artificial lysins because it's the molecules modified by adding some special addition, and this modified molecule show increased activity against gram-negative bacteria. And artilysin can be used to treat opportunic infection, gram-negative drug-resistant strains of Pseudomonas aeruginosa and Acinetobacter baumannii. Here you have some picture from the microscopy, and after 21 minutes after added to the culture of Pseudomonas aeruginosa, cell was completely broken. And to sum up my presentation, pros and cons of using murine hydrolysis. On this plus side, we can find that these enzymes are offensive against gram-positive antibiotic-resistant bacteria, and the mechanism of action murine hydrolysis are different than mechanism of antibiotics. So the current antibiotic-resistant mechanism don't play with the murine hydrolysis. Murine hydrolysis are well known and safe, and they are effective even in the small doses. So we can avoid the immune response in patients. And on the minus side, we can find that unmodified hydrolysis, murine hydrolysis show weak activity against gram-negative bacteria. They have some activity, but they is weak in comparison to the gram-positive. And production is relatively expensive, for example, to comparison to the bacteriophages. And to total sum up, I would like to return to my title question. Can murine hydrolysis replace antibiotics? I don't know. Times will tell. Thank you for your attention.
Thank you very much for an interesting presentation. Do we have any questions?
How does murine hydrolysis affect the microbiome? Because recently there's more and more information about how the microbiome affects all kinds of activity inside the organism. So doesn't murine hydrolysis damage it?
Yes, of course. Like the situation is similar like in antibiotics. Antibiotics cause on the pathogen and natural microflora. So yes, it can be a problem. But another example of using murine hydrolysis is to treat the skin infection. So in this context, it will be much better, and the microbiome will not disrupt.
Thank you.
Any more questions?
I have a small question because do you know anything about experiments using the murine hydrolysis to test how quickly or whether it's possible for bacteria to develop resistance?
It is a very great question, because yes, this experiment exists. I don't show it in my presentation because I thought it's too more detailed. According to the author of this publication, murine hydrolysis do not have the resistance mechanism even in 30 culture passages, how to say in English, you know, they made some experiment. The culture of bacteria, the sublethal dose of murine hydrolysis, and next testing the potential resistance, and no, they didn't find the resistance. And this is one of the another plus side of the pros of using the murine hydrolysis, that they don't generate the resistance. Of course, I can add some other details that not every murine hydrolysis will be good for every bacteria, because murine of each species of bacteria is different. And for example, my agent of my PhD study, Listeria monocytogenes, has modification into the murine, some glycosylation of some compounds in murine, and it's due to that this bacteria is natural resistance for some kind of, for example, lysosome.
I saw another question.
Okay, but on what kind of bacteria were the resistance tests done? Because of course the time of the development of the resistance will be different for another it will differ from bacteria to bacteria, and also the number of passages doesn't exactly reflect the number of bacterial generations, because every bacteria also the time is different. For example, Salmonella divides every 20 minutes. But there are bacteria that divide every 4 or 5 hours, so it depends on bacterial generations.
I thought I have this extra slide on this presentation, but now in this version, these scientists tested some kind of virus-related murine hydrolysis, and I think 3 different antibiotics. And after these 30 passages, all of the 3 antibiotics generated the resistance, and murine hydrolysis didn't. You ask about how?
The genus of the bacteria.
I don't remember, sorry.
Okay.
Anyone else would like to ask a question? If not, then thank you very much. And this is the end of the session, so now we have a short coffee break.